Garlic? Really? It disrupts biofilms like a weapon, just like in the movie. Iron Schwarzenegger.
In this video, I'm going to be talking about biofilms, as well as how garlic has an effect on biofilms. So, garlic contains organosulfur compounds such as allicin and diallyl sulfides. And these compounds, they can react with cysteine thiol residues and also disrupt disulfide bonds and proteins. And on biofilms, many of these bacterial biofilms, they depend on thiol-rich adhesion proteins. Also, biofilms depend on fibronectin binding proteins, especially in Staphylococcus species, which is a bacterial strain. But garlic sulfur compounds, they can inactivate quorum sensing enzymes, which I'll explain that later. And also, garlic sulfur compounds destabilize fibronectin mediated adhesion, increasing biofilm permeability. Now, as I said earlier, that the biofilms in bacteria, they depend on fibronectin binding proteins. But garlic sulfur compounds, they destabilize these fibronectin proteins, therefore destabilizing biofilms too. Okay, so there's this thing called a quorum sensing, and certain bacteria, they need the quorum sensing signals in order to form biofilms. But there are some things that can modulate the quorum sensing, and one of them is asparagus, and its extracts have been shown to inhibit quorum sensing. This means that it can reduce the chemical communication bacteria use to coordinate biofilm formulation. This effect is typically more pronounced against gram-negative bacteria or lipopolysaccharides caused by gluten ingestion, and they also rely heavily on quorum sensing systems. And by interfering with quorum sensing, asparagus compounds can reduce biofilm density and stability, making bacterial colonies less organized and more susceptible to antimicrobial agents. Now, it's critical for these biofilms to be reduced because the immune cells, macrophages, they can't really engulf or do the phagocytosis on bacteria and pathogens and cancer cells that are using biofilms as protection. So, bacteria, pathogens, and cancer, they use biofilms as a shield from phagocytosis, because that's what macrophages use to clean up debris such as pathogens. Because if they're not cleared, then they can cause inflammation, cancer metastasis, and other problems. So that's why macrophages are supposed to go around cleaning up pathogens and cancer cells, but they can't do that if the biofilms are not degraded. So there has to be a way, or there has to be mechanisms or ways to support and to help out macrophages so that they can clean up cancer cells and pathogens and dying cells. And degrading biofilms with garlic and asparagus is a great way to help out the macrophages of the immune system. It has been shown that higher concentrations of asparagus extracts usually leads to more significant disruption of biofilm structure. So that's critical to help out the macrophages. So asparagus can act as a prebiotic for the gut bacteria because the A gut bacteria needs prebiotics to grow so that the gut bacteria can serve and support the immune system, but also as an anti-biofilm potential. And asparagus can synergize with probiotics and other plant compounds like turmeric or pineapple that shift toward saccharolytic species to limit biofilm formation by harmful bacteria. So asparagus combined with pineapple makes a great team because asparagus weakens or prevents the biofilm formation, leaving cancer cells exposed to pineapple's effects because pineapple cleaves CD44, CD62L. What that allows or enables is the infiltration of the cancer cells with T cells. So the T cells ends up infiltrating and attacking the cancer cells thanks to a pineapple enabling the access of the T cells into the ECM, or short for extracellular matrix, and the cancer cells will be weakened and exposed if they don't have the biofilms protecting and surrounding the T cells, thanks to garlic, allicin, and asparagus compounds or extracts removing the biofilms from the cancer cells. Okay, so quick recap. Bacteria, they use quorum sensing signaling as a way to upregulate biofilms, but asparagus extracts are known to reduce these biofilms. Also, higher concentrations of asparagus extracts leads to even more degradation of biofilms. Biofilms also rely on or depend on thiol-rich adhesion proteins and fibronectin binding proteins, but garlic sulfur compounds such as allicin destabilize the fibronectin adhesion as well as destabilizing the thiol-rich proteins, therefore degrading the biofilms. The macrophages are responsible for cleaning up uh, cancer cells, pathogens, debris. If they are not cleaned up, they will cause inflammation and other problems. But the macrophages, they cannot use phagocytosis, which is an engulfing sort of attack or action or mechanism that macrophages use to either clean up debris and dying cells or to eliminate or destroy cancer cells and pathogens. But macrophages cannot do this if there are biofilms uh, surrounding or protecting debris and pathogens and cancer cells. But thanks to garlic and asparagus, uh, the macrophages are able to clean up pathogens and debris and destroy cancer cells because asparagus and garlic, they get rid of or degrade the biofilms. Okay. Uh, Asparagus and garlic are also a good combination with or can team up with a pineapple. Pineapple is a protease fruit, which makes it different and unique compared to other fruits because other fruits are not proteases the way pineapples are. And it has a cutting or cleaving effect on the ECM, making it less rigid and more loose for for immune cells to enter and attack cancer cells. Uh, of course, the cancer cells cannot have the biofilms surrounding or protecting them. That's why asparagus uh, and garlic removes the biofilms, leaving the cancer cells vulnerable and exposed to the attack of the T cells. And pineapple also cleaves CD44, CD62L, so that the immune cells can infiltrate and home in onto the cancer cells. That's why pineapple is a good combination with asparagus and garlic. So that's it for the video. If you want more future videos, then like, share, comment, and subscribe. Keep it locked in here on my channel, and I'll see you in the next video.